First, we looked around us and saw SARS-CoV-2, which had already had at least 3 genotypes. Some papers suggested that genetic variants could impact ways that doctors should cure patients. But of course, we could not be sure at that point, so we asked for experts' opinions. We asked researchers, health workers and officials about the possibility that coronavirus genotypes could influence the treatment choice. It turned out that there was no strong correlation between SARS-CoV-2 genotypes, cause symptoms, and treatment. We had to reject our first idea, but we did not give up. We made a list of other RNA viruses suggesting that virus genotype detection could be useful for some other diseases. And we thought of hepatitis C even before the Nobel Prize was awarded for its discovery. Why? There is no HCV vaccine. There are several different genotypes of the virus. There are quite a lot of therapy strategies. Some are cheaper, some are more expensive, some are pangenotypic, some are not. High risk of progressing to chronic hepatitis and cirrhosis. Reinfection possibility. Altogether, seems to be quite a problem. Moreover, in Russia there is one special recombinant variant of HCV. It is common and is often mistaken with other genotypes. At that point of our project, we not only spoke to experts in epidemiology and medicine, but also to patients. We contacted a non-profit patients organization Together Against Hepatitis. Patients organization highlighted 3 big problems with viral hepatitis: lack of informational support, lack of insurance-covered tests, and insurance-covered medicines. This point of view was confirmed by other experts, including health workers and officials. The meetings with the experts showed us that even though there are pangenotypic medicines, the HCV genotype still influences the therapy choice, and that we could help by creating an easy-to-use and relatively inexpensive virus genotype detection platform. But we also could help by starting an HCV awareness campaign. In collaboration with a non-profit organization, our team has started a series of informational posts on social media, and we are looking forward to the future possibilities in this field. Later, we asked BioCat biotech company experts for feedback on our project. That was a fruitful interaction. Following experts' notes and advice, our team changed the concept of the test platform not once. Teammates reconsidered both the molecular technologies used in the platform and the hardware to implement it. It was inspiring to watch how the concept was changing, following the patients' and health workers' demands and experts' opinions.
The choice of the object for detection wasn't the only question. We had to propose a suitable detection method. The main idea was simple. Take the blood sample, put it into the detection box, and look at the results. But how to make it fast, easy to use, and specific? We consulted with our PIs, and they told us about the possibilities of using CRISPR-Cas systems, for which the Nobel Prize 2020 in chemistry was awarded. During our project, we designed a detection system that requires Cas-X protein as a key component of our nucleic acid biosensor. In the end, we came up with the following system. A blood sample is supposed to be collected by junior medical staff in any medical institution. Virus from a patient's blood is inactivated at 95 degrees Celsius by the Hudson method. Next, RNA is converted into DNA and amplified with the help of the LAMP method. In the presence of genetic material from a certain genotype of the pathogen, Cas-X cuts the reporter molecule, labeled with biotin and the fluorophore from opposite sides. The resulting solution is placed on a test strip with 2 binding zones. The uncleaved molecules will bind to the control site on test strips, while the cleaved ones will bind to the second site. Results can be visualized with the help of a fluorimeter. Two bands on the test strip indicate the presence of a specific viral genotype in the sample. One indicates its absence. The only equipment required is a simple thermostat, since a fluorimeter is a part of our biosensor. We started with an idea of a detection system for SARS-CoV-2 genotypes and made a list of single nucleotide polymorphisms for it. We worked through the literature about SARS-CoV-2 mutations and the division of the virus into clades. We used BenchLink to map SNPs to sequences and find potential protospacer adjacent motifs, or PAMs for short. Then, taking into consideration the experts' feedback that we got during the human practices work, our project switched to HCV. We also made our own alignment of 17 genomes belonging to the most common genetic variants of HCV and selected SNPs for future development of guide RNAs to target Cas6 protein. Then we created a detailed kinetic model to define optimal conditions, reaction time, and potential sensitivity of our detector. The model played a vital role in the development of our project because we wanted to make sure that our proposed system is capable of detecting viral RNA at low concentrations in an acceptable amount of time. Most importantly, with the help of modeling, we understood that we can reduce the level of RNA degradation and thus increase signal of fluorescence. We implemented an additional heating step, Hudson method, into HaploSense. We also found that the system can detect nanomolar concentrations of viral RNA, and the overall response time is potentially about 1 hour, which is great, although it will depend on the hardware. Due to modeling, we improved the detection system and set the initial parameters for its work. But before we continue our work, we need to return to the lab, where we can extract Cas6 protein and test our detector. Developing our project, we primarily aimed to create a test system that could be used in Russia. Existing test methods like real-time PCR and sequencing are time-consuming, require qualified laboratory staff, and expensive equipment. By creating a fast, easy-to-use, and relatively cheap test system, we wanted to make HCV genotyping more affordable for infected people in Russia, to make their treatment more effective. We also thought about safety. As soon as HCV testing requires collection of blood samples, we did not and won't use alive viruses, their parts, or contact infected persons. When implemented in the real world, blood samples are supposed to be collected by medical staff only and handled with precautions. In any case, potential pathogens from blood will be inactivated in the first step of the detection by the Hudson method. Though our biosensor involves complex macromolecular structures, if the device is damaged, the components could not bring any harm to the environment or a human being. First of all, we are planning to validate and patent our system. HaploSense might become the first CRISPR-Cas-based testing method in Russia. Thus, we most probably will meet difficulties on paper step. We also hope to implement our test system into medical institutions. And we hope that it would help a lot to hepatitis C patients. Apart from this, our test system might become a universal tool for the detection of any RNA viruses genotypes.